Welcome to the Inspiring Minds coaching, and let's begin with the microbiology. And here in my last video, I talk about the bacterial structure, and you can see there in that video, I told you about the important structure on only their names. And now, after that video, I told you that later I will take their structural component and function so that you can go through the questions and you can solve all questions regarding the structure of the bacteria. Okay. So as in my last video, just I told, and here just I am giving you a quick idea that bacteria, the Monera from the Monera kingdom, they have body, appendages, and a special structure, and that special structure was the spore. Okay, and body was containing the that cytoplasm and inside cytoplasm things and nucleus. Appendages were the capsule, cell wall, fimbriae, and pili. When you go through the Kaplan notes and you try to understand the step one things, either in elsewhere or in Kaplan, what you will find that there is two specific type of classification. The first one is essential, and the second one is non-essential. See guys, that essential bacterial structure component is basically, what does it mean? It means that these structures are important for the bacteria itself. We are not going to talk about the pathogenetic activity of the bacteria. Here, these structures are very important for the survival of the bacteria, right? That's why you don't have to take it like this way, like essential structure will contain the pathogenic activity of the bacteria. Okay? So here, the first one, essential structure of the bacteria contain the first one, cell wall, second one, plasma membrane, third, ribosome, 70s ribosomes, and fourth, very important, nucleoid, and then mesosome, periplasm. And non-essential structure contain capsule, pili, fimbriae, spores, and plasmid. These structures found in the bacteria, but they don't have the benefit effect for the bacteria itself. That's why they are in the non-essential category. The essential part of the bacteria is very important for their survival, reproduction, and also from one generation to another generation, like their other functions, which I will tell in the bacterial genetics. So just take one by one. Cell wall of the bacteria. Just make it sure that whenever there is, I think that you all guys know that cell wall is the specific property of the plant cells. But here that cellulose containing, but here cell wall, bacteria contains cell wall. And what about animal cells? Animal cells, yes, right, they have the cell membrane, lipid bilayer cell membrane. Everyone knows. And when we talk about the drug activity or any kind of transport, then there we always study about the cell membrane activity. Okay, cell membrane and medium. So even also in case of the body circulating fluid. Here I am taking one by one, and I will talk about their component, this part, and here is component, and here their arrangement and their function. Why only I take these things? Because these are the exam based questions, and every point where I'm gonna make the star, they are important according to the exam point of view, especially when you deal with the assembly questions. Because many times what happens, we don't know the proper meaning of that, and somehow they said their non-essential structure of the bacteria have been they did not found in the staining medium. So that simply means you have to make it clear in your mind. Capsules, pili, fimbriae, and spores and plasmid, they were not in the staining culture, or they may say that they were present. So on the behalf of them, you can just go through that clinical diagnosis, and you can say that yes, that structure is not found in that bacteria medium. That's why. So you can say you can just guess that yeah, that is that kind of bacteria. It's very important. It's very important to make it clear, because when we go through the clinical in my last video, last video, I talk about the lab technique of the bacteria, or you can say the systemic bacteriology, and there we study about only few staining technique. They were basically based on their cell wall structure, or there were some on the behalf of their capsule structure, and some of them because of their endotoxin secretion. All these things are important on the bacteria point of view. But when we talk about the laboratory diagnosis, they have the special impact over the body, right? So the first one, just taking that, the first one is cell wall. This very famous and very important topic on the behalf of essential structure of the bacteria, the cell wall. Cell wall is composed of the peptidoglycan layer. Keep in mind peptidoglycan. Okay, and as we all know that bacteria have the two kinds of categories. The basic one, the gram positive, gram negative. So here the gram positive bacteria have the thicker cell wall. Okay, and they have outer surface fibers and structure. Okay, I have mentioned here. And these the component of the cell wall is the major component is peptidoglycan. Okay, in all bacteria, gram positive, gram negative in both. They are cross-linked arranged. If you don't know them, then go through my previous video. There I told the cross-linked arrangement. It was like one slide, one ladders arrangement. And exams point of view, if I tell you that everything is not important for you to understand, because somehow when you watch my first video, and there explain most of the things. Okay. Second, about stain, and then later I talked about the few morphological and physiological property. So I'm not going to tell every time their basic and by default functions. Here is specific things only exam point of view. Don't waste your time, just go through it. That the peptidoglycan layer, the cell wall composed of peptidoglycan. So just to make it short, cell wall composed of peptidoglycan in gram positive, gram negative both. Gram positive and gram negative both. Okay, they are cross-linked arranged in the gram positive, in my previous video I told, and in the gram negative they have the single layer. Site of action, here I talked about the specific drug, because as a medical student, it's not important for you. It's not necessary that every time they will ask you only and only go through the theoretical questions. They may ask you about applications. The site of action of those drugs are the penicillin and cephalosporin. Okay, penicillin and cephalosporin. This is antibiotic, very famous antibiotic. So keep in mind that penicillin and cephalosporin, these antibiotics have effect over the peptidoglycan chemical component of the cell wall of the bacteria, and that peptidoglycan layer found in gram positive and gram negative both bacteria, but they have major impact over the gram positive bacteria. Okay, so the gram positive bacteria, and also there is outer layer of gram positive bacteria is lipid A. Sometimes it's a lipopolysaccharide A, and they produce endotoxin. Toxic pathological activity, which they cause the pathological activities in the host cells. They may be animal, any kind of animal, human. So there they have endotoxin and exotoxin. When you talk about the endotoxin, they have gram positive bacteria have the outer layer lipid A containing, they secrete endotoxin. Okay, the gram positive surface fibers, they are very important because from the schooling period we all know that gram positive bacteria containing one special kind of molecule or chemical substance that is only and only found in gram positive bacteria cell wall, that is teichoic acid. And I'm telling it the third time because it is going to be asked in the most of the questions. This is a structural component. And one thing more in the gram positive bacteria surface, that is polysaccharide layer. They are very important in the clinical diagnosis. They're not important, but whenever you have a question and it says that gram positive bacteria only contain the teichoic acid, that's absolutely wrong, because they also contain the fibers. In my last video I told you about the fibers. Here their chemical nature is polysaccharide. Okay, so the first one, cell wall, is completed here, and the second one, plasma membrane. The plasma membrane, remember that plasma membrane is also found in animal cells. Even you can say this is a universal truth in case of the any kind of animal cells. or any kind of living cell. In almost there is a plasma membrane here. Its concept says that plasma means inside of that fluids, and that membrane is the outermost boundary, right? So here you can say that the plasma membrane of the bacteria, it's very important, but not more important than priority wise. Cell wall is more important. And the second one, they contain lipoproteins. They don't have lipopolysaccharide. This question have been asked and many times we did wrong. This is lipoprotein. Okay, lipoprotein plasma membrane, and they are the bilayer. One thing more, bilayer cell wall membrane. If you remember that those bilayer structure I have made in my previous structure, it was like this way. Okay, lipoprotein bilayer, and what do they have, or you can say their function? They contain oxidative and transport enzymes. Okay, oxidative for the respiration of the bacteria and transport for the material. Okay, so just keep in mind oxidative and transport material found in the plasma membrane of the bacteria. And now the next one, the third, that is ribosome. I think everyone knows that 70s ribosomes found in bacteria. Those who don't know, just make it last time, because every time you will find difficulty if you don't know these basic things. 70s ribosomes, and they contain 50 and 30. It was NEET examination question. So now don't expect that they are going to ask that how many ribosomes are there. Ribosome subunit in the bacteria, because it is a very lower standard question. So they contain 2 ribosomal subunit, 50 and 30. Site of action is the chloramphenicol, erythromycin, tetracycline, and aminoglycosides. These all are the drugs, or you can say the antibiotics. Antibacterial agents: chloramphenicol, erythromycin, tetracycline, aminoglycosides. Okay, once more, here if you see everything, so you need to remember them like just you need to remember, you have to remember 1 or 2, that these are the enough. But I will give you can just have a 1 letter C, E, T and A. Okay, like kit. It's not cat, it's kit. Okay, so the 4 drugs which directly act over the ribosomes of the bacteria, these are the chloramphenicol, erythromycin, tetracycline and aminoglycosides. And letter 3, I think you all know: nucleoids, mesosomes, periplasm. If you don't know, in the bacterial genetics, I will talk about the nucleoids, because there is a bacteria reproduction and how they form another bacteria. And in the mesosome and the periplasm somehow it is you can say, in my previous video I told you about the periplasm, and once more I want to tell you that this is the bacterial cell. There is a nucleoid material, there is a plasmid and cellular organelles and 70s ribosomes, the outer cell wall. Okay, and there is periplasm, and there is the inner one after just cytoplasm, that is periplasm, and then there is plasma membrane, and then there is cell wall. Just keep in mind that you don't have to make it so much complicated while study, because more simpler you study, the more simpler, it will be long time retain in your brain. Okay, once more I am starting from the first, the bacterial structure and function component. The bacterial structure, its function and component have been talked in this video, and I told about the bacteria contain the body, appendages and special structure, and those special structure of the spores, they are very important for the survival in extreme temperature for the bacteria. And appendages and body part, most of the part of the appendages, they are the non-essential part of the bacteria. And when we talk about when we talk about body part, they contain mainly essential part of the bacteria. The essential part of the bacteria contains cell wall, plasma membrane, ribosomes, nucleoids, mesosomes, periplasm. They have their property. They have very importance for the survival and survival and continuity of their life in essential structure of the bacterial structure. And the non-essential, they contain capsule, pili, membrane, spores and plasmids. They are non-essential for the bacteria itself, but they are very important for the pathogenicity. So you don't have to be confused that whenever if there is non-essential structure have to be removed. So what will do bacteria? Basically bacteria will survive, but they will be unable to cause the disease, because most of the disease impact is caused by the non-essential structure, but not all. Okay. And the cell wall component is peptidoglycan, and peptidoglycan layer present in gram positive, gram negative both bacteria. Gram positive, gram negative both bacteria. It is a universal thing. And peptidoglycan is cross-linked arranged in gram positive, and the site of action of the drug, that is penicillin and cephalosporin antibiotic. And if you talk about the animal, so an animal cells, like that is lysozymes, when we contain any kind of bacteria, so we have cellular lysozymes, hydrolytic enzymes, and they are able to act over the cell wall of the bacteria, and they degrade those bacteria, and we are safe. The gram positive bacteria have the outer layer, one more, that is composed of the lipid A, and they secrete the endotoxin. This is a toxic compound which is secreted for the pathogenic activity over the animal cells. And the gram positive surface fibers, they have the very important teichoic acid, because it has been asked many times in questions. Sometimes what happened, they simply say teichoic acid containing bacteria. You have to make it clear they are the gram positive bacteria. Okay. And when we talk about the cell surface fibers, there is not only teichoic acid but also polysaccharides present. It has been asked in assembly question that was like teichoic acid and polysaccharide, and teichoic acid only. So don't have to be confused, you have to take both, this also and this also. And teichoic acid is especially the component of the gram positive bacteria, but polysaccharides, they are very helpful for the clinical diagnosis. Now the second one, plasma membrane, they are the lipoprotein containing bilayer, and they contain oxidative or transport enzymes, and oxidative and transport enzymes found there for the respiration of the bacteria and material transfer of the bacteria from the outer external environment to the inside. And ribosomes, the 70s containing ribosomes, 2 subunit 50 and 30. The site of action is antibiotics, the Kate, that immunomics you have to remember, the chloramphenicol, erythromycin, tetracycline and aminoglycosides. Nucleoids, they are the double stranded and spread based structures which is found inside the bacteria. Mesosomes, they have the very important function in when we talk about the enzymatic activity. Periplasm, the outermost layer of the cytoplasm that contain the most of the hydrolytic enzymes. The very important, that is question asked, that is beta lactamase. Okay. So one thing, before finishing this video, I would like to make it clear that antibiotic which act over the cell wall surface of the bacteria, they are the bacteriostatic property. Okay. When we talk about the bacteriostatic bacteria, antibiotic, bacteriostatic, and when we talk about the bactericidal. When we talk about the bacteriostatic, so simply when those antibiotic or those drugs which act over the cell wall, these are the bacteriostatic bacteria, and those drugs which act over the ribosomes, or you can say the protein forming component of the bacteria, so that they can't survive. These are the bactericidal drug. They kill and they stop bacteriostatic. Okay. So everything is finished here, and all important things I have mentioned here. So don't expect that I never write the whole theoretical part, because I personally don't like to study one by one everything, and just I'm trying to give you the most important content of the bacterial structure of the essential part, and the next one that is very interesting in the pathological point of view and also drug activity, that is non-essential. Their gram stain, staining property, lab diagnosis, and also acid fast. All these things, these are the based on the non-essential structure, and the pathological activity like Neisseria, Clostridium, and gonorrhea and Haemophilus influenza. These all things, they will be in the, their activity will be in the non-essential part. So hope you guys like this video. Like, share and subscribe, and later in the next video let's talk about the non-essential part of the bacterial structure. Thank you.